Oh, Lauren, can I pull you outside? Sure. Okay, so welcome to class. So enrollment is at 7 people, so we're actually going to have a lot of fun this semester, and the grading is going to, in the past where I've done, sorry, let me put the microphone on. The past where I've done iClicker grades, we're going to do journal club discussions on interesting papers. So you'll learn a ton. So how the class will be structured is every class we're gonna spend the first 30 to 45 minutes doing a journal club discussion. This is not on, is it? Okay, so for next class, for our journal club, we're going to cover MegaHit, an ultra-fast single-node solution for large and complex metagenomic assembly via succinct de Bruijn graph. So we'll go over this more at the end of lecture. So for the participation grade portion of your class, I'll send out a list of questions. You'll need to read the paper, and then there's no right or wrong answer. If you come to class and participate in the discussion, you get full points there. And I'll bring in some of my PhD students to make the discussions more interesting. And then finally, we're going to have 2 graded homework assignments as per our syllabus last time. Since the class is small, we've switched it up to make it more interesting. The first homework assignment for the first half of the semester is on building a computer vision pipeline to handle compression of large-scale medical imaging. So what we're going to have you do is we're focusing on a lot of skill sets for deep learning using stacks that industry actually uses. This is a little different from what Google would actually use, because if we were doing multi-GPU training, they have their own pipelines like JAX, but we're actually we've given you a lot of starter code and explanation of how a variational autoencoder works. We've essentially asked ChatGPT to solve the problem, and then everything that ChatGPT got correct, we've given you that 70% of the code, and you need to just fill in the rest. So we have instructions about how to do this on Collab, what all the parts for the deep learning assignment are, and then how you get points for things. And we'll go over this all in more detail. For extra credit, if you don't want to take the midterm, in a variational autoencoder, we have, it's just an autoencoder with a Bayesian spin, in that we're using our variational parameters to sample from a distribution in our latent space to feed into our ResNet that after we compress it is uncompressing it. We have our encoder and decoder. So if you don't want to take the midterm, you can come up with an interesting project that takes a real medical imaging database and uses this latent space to do a different downstream task other than reconstructing the original image. We're going to cover in the next couple lectures all of the math and all of the libraries that you need to know to accomplish this assignment. We'll post it now and make it due around the middle of the semester so you can get a head start on it. But if you're a master's student and a lot of this is new to you, there's no reason to be scared. So on to our lecture. So today we're covering a brief overview and what you need to know for the course in molecular biology and genetics, programming and stats, and then I'll go into a more detailed explanation of the first homework assignment and the journal club assignment. I'm also going to post, it's become much more challenging to post to grade non-individual assignments with ChatGPT. So we're going to distribute a lot of our previous homework assignments that we graded that are just for your educational enrichment. So some of the content will be covered on the midterm and final, but we're not going to assign a grade on the homeworks. So I'll go over some of those homeworks too. Thank you to students who pointed out the syllabus and Echo 360 issues. I will work with the TAs to get everything updated after class today, including this. Okay, last class I promised that we would cover when office hours occur. So I'll have office hours immediately following class and Mondays by appointment. The TA is no longer Mohammed Nasser. I need to fix that slide. Jay Bukas Verla. I'll be distributing his email to schedule by appointment office hours in SEER 426. As a note, SEER 4 has some biohazard and radiation laboratories, so card access is controlled. It's totally safe, but you will not be able to access the TA office hours without a previous appointment because of the card access restrictions in the building. So a key idea, so this is also an outdated slide because we have replaced our quizzes with our journal clubs. But a key idea in the course is that we're measuring different levels of the central dogma and then computationally parsing those. Almost every year there's a short answer question to make sure you understand the central dogma of how DNA is transcribed into mRNA, which is then translated into amino acids, which are made into proteins. And so we can measure DNA, mRNA with sequencing, and do more advanced imaging and mass spec-based measuring approaches to measure protein. Genetics and genomics are closely related. Genetics, we could say, is the study of how traits are passed in a hereditary fashion in population, and genomics are how we use high throughput tools to measure genetics. So to understand all of this happens, it's really useful to understand some cell structure so we can think about how different levels of the central dogma are orchestrated. So here I've shown an animal eukaryotic cell, which is different than a bacterial prokaryotic cell. We have a nucleus where all of our DNA is stored. We have a lot of different cellular components that do different things. And then we have our cell wall. And so, for example, ribosomes are involved in translation. So our DNA is in the nucleus, and then inside the nucleus, our DNA is we can view DNA as just a double helix of 2 strings with an alphabet of 4 characters for all of our, since we're all computer scientists. And then that's copied to mRNA, which is a slightly different alphabet. We replace T with U because of some biochemistry beyond the scope of this course. And then the mRNA leaves the nucleus and travels to our ribosomes. And then our ribosomes use this code of 3 nucleotides to make codons, which correlate to 20 amino acids, which are assembled sort of like Legos to make proteins. So the algorithmic relevance here is we say in genomics, we're measuring or quantifying DNA in the nucleus of the cell. In transcriptomics, we're measuring mRNA that's transiting between the nucleus and the ribosome. And then proteomics is measuring protein. And if we think about what techniques we use for these, in measuring DNA, this is a lot of graph algorithms, like De Bruijn graphs, and thinking about some string algorithms like shortest common superstring to assemble short substrings of DNA we measure into longer contiguous strings. Transcriptomics is more about dynamic programming algorithms and how we think about taking simple string-based algorithms like the Needleman-Wunsch algorithm and the Smith-Waterman algorithm and scaling those so we can run 700 million at a time. And then proteomics is measuring protein. We can't directly sequence proteins right now, so these are mostly histology and imaging-based approaches and mass spec approaches. What exactly is a gene? So we can say that we have our mRNA so if we go to panel B here, we see the double helix where we have complementary nucleotides A, T, and C and G that form DNA. And this is a really efficient compression system because we actually, in a eukaryote, have 6 frames of reference. Because we can think about this as an array of characters, and we can say we have our first codon TAC. We can also introduce an off by 1 error and have a totally different frame of reference. And we can do an off by 2 error, etc. And then, so this can be translated into many different strands of mRNA, which is turned into proteins. So when the ribosome is reading our protein, we have a start codon AUG, and that all the different permutations of substrings of 3 characters correspond to 20 amino acids. And so when we code M, our start codon, and then we have AUC, which is I, etc. And we add these amino acids in a chain to each other, and then they fold into protein structures. And protein is what makes up, these proteins are what make up all life. So the genetic code in DNA encodes what our proteins will look like. And so now let's step back into the 1800s and talk about Gregor Mendel. So Mendel was a monk from 1822 to 1884 in a monastery in Austria. And despite being part of the clergy, he really wasn't that interested in his religious pastimes. And while studying scripture in his monastery, he became the father of modern genetics. And he was obsessed for decades with breeding pea plants. And he discovered Mendelian traits and a system of rules to define observational experiments. And that the key discovery he made is that parents have different versions of genes that can be passed to offspring. So let's step back and ask, what is genetics? So genetics is the study of how traits are inherited in populations, specifically how traits are passed to offspring. And this can be really complex. We can have autosomal dominant inheritance, where you just need one copy of a gene to have an affected child, and this is things like Marfan syndrome, which affects your tendons and gives your joints a really weird appearance, and Huntington disease, where your brain degeneratively turns to mush at around age 40. We can also have autosomal recessive inheritance, where you need a copy of the trait from each parent in order to get to the disease. So in this case, if a parent's a carrier and the other parent is unaffected, some of the children will be unaffected and some of them will be carriers, assuming Mendelian inheritance. And so this is diseases like cystic fibrosis, mucus accumulation in the lung, and sickle cell disease. And so in Mendelian genetics, we think about dominant versus recessive traits. And an example of a recessive Mendelian trait in human populations is attached earlobes. So if you look at your earlobes, at the bottom of them, they're either attached to your face or not. So you can see that I have attached earlobes. That little piece of flesh is attached to my cheeks, like the figure on the left, or you can have detached earlobes. And so having attached earlobes is a recessive trait, meaning that to have attached earlobes, both of your parents need to have it. And if neither of your parents or one of your parents has attached earlobes, you would have detached earlobes. And so these types of traits were discovered by Mendel. If you're interested in this, there's some additional excellent resources in the 6th edition of Watson's Molecular Biology of the Gene. So when Mendel was breeding peas, here's an example of a Punnett square from one of Mendel's breeding experiments. Say for his parent generation, he had a group of yellow, round peas and green wrinkled peas. We can see that we've denoted the yellow round pea as capital YYRR and the green pea as lowercase YYRR. So in this case, yellow and round are dominant traits. If a pea has any capital, so we can see that one of these Y's come from each parent. If there's any capital Y, the pea will be yellow, and the pea will be green and wrinkled only if it's all lowercase. So we can see that when we breed these peas, in our first generation, if you look at the combinatoric possibilities of crossing all possible gametes from the parent generation, we'll always get yellow round peas that are carrying the recessive genes, YYRR. So if we then go and breed the mixed Y, mixed R, F1 generation with itself, we make this table of 16 possibilities. And so this is the observational experiment that Mendel did in his monastery. And he looked at all 4 possible combinations that could be passed from this parent. It could be Y, capital R, lowercase y, capital Y, lowercase r, and then all lowercase. And then the second parent is the same, since we're breeding genetically identical pea plants. And so we can see here that I've drawn out the genotype of all the offspring peas lettered inside it, and then the phenotype of what the P actually looks like. So we say genotype is the actual genetic traits, and the phenotype is the appearance. So you can see that genotype and phenotype are not always equivalent to each other. A P that's capital Y, capital R, R appears physically the same as a P that's capital Y, lowercase r, capital R, lowercase r. And that the genotype is different, but physically and functionally these P's are the same. Their offspring would have different characteristics from each other. So if we grid these possible offspring from breeding this F1 generation of P's, I have this more clear here. We can say that we have 9 round yellow P's, 3 round green P's, 3 wrinkled yellow P's. So you can see that we only have 3 wrinkled yellow and only 3 round green, and 1 wrinkled green P. And we have a 9 to 3 to 3 to 1 ratio in this. And by breeding thousands of pea plants, Mendel discovered that this ratio is explained by the law of large numbers. This ratio is roughly fixed regardless of the number of pea plants we breed or the number of offspring we have. And so this is really the foundational work that you need to know from biology to think about these problems computationally. Because once we move to human genetics, things become much more complex, where we can have things like, so just as a starting point, are there any questions so far? As a starting point, human genetics is not Mendelian. There's some Mendelian traits, like attached earlobes, but we have way more complex interactions between genes determining traits in human populations. For example, we can have pleiotropy, which is where a single gene controls multiple traits. The expression or presence of one gene can control trait A, trait B, trait C, and trait D. An example of this is the disease sickle cell anemia, which arose in populations of African ancestry, which is an inherited red blood cell disorder that causes multiple pleiotropic phenotypes. So one mutation in beta globin causes diseased red blood cells, vascular damage, but it also confers resistance to malaria. So evolutionarily, some genetic diseases likely had advantages that caused, in terms of evolutionary pressure, that caused them to arise in populations. Before modern medicine, getting malaria in certain regions of sub-Saharan Africa could be a death sentence. Hence why these selective pressures likely selected many of these mutations. We can also have polygenic inheritance. Multiple genes control a single trait. So we can have a list of genes, gene A, gene B, gene C, gene D, that control one trait. An example here is height. Differences in height across the population is caused by polygenic inheritance. It's unlike attached earlobes, there isn't one gene that says you're tall or you're short. There's a collection of a few hundred genes that, when you look at their additive effect, determine your height along a distribution. So human genetics is much more complex. Are there any questions so far? Okay, so DNA, we'll now talk a little bit about DNA, genome sequencing, and graph algorithms, programming introductions, and statistics. DNA was discovered in the 1950s. I believe the seminal Nature paper was in 1954. by Watson, Crick, and Franklin. And they discovered it through X-ray crystallography, where the genetic material is these nucleotides in 2 strings. If you have one of these 2 strings, you can recreate the other half of it computationally. And we'll talk about some applications of that that fits in the sugar phosphate backbone. And we have purines, adenine and guanine, and pyrimidines, thymine and cytosine. Cytosine pairs with guanine, and adenine pairs with thymine. And the DNA follows this double helix structure that's wrapped into chromosomes. And we have 20-something chromosomes that we'll talk about that are these. We pack our DNA into this structure called chromatin, and when a gene is expressed, the chromatin unwraps in the chromosome, and we express that gene. We'll talk more about how we computationally think about this in the GWAS example in just a second. When we go to sequence something, there's a few platforms we can use. A decade ago, there was closer to 6 or 7 platforms, but a lot of genome sequencing companies like Ion Torrent and the Roche 454 have all been acquired. So for most of the last decade, there was 3 main players: Illumina, Oxford Nanopore, and PacBio. Illumina and PacBio, Illumina acquired PacBio, so there's actually very few companies that are doing genome sequencing. I made one notable exclusion here. The Beijing Genomics Institute, which despite its name is located in Shenzhen, has a nanoball technology that uses different IP than either Oxford Nanopore or Illumina. So how Illumina works is we take our genome and we split it into these tiny fragments. And then we put adapters onto these fragments. And you can see in the photo here that those adapters, we attach them to a glass plate. And genome sequencing technology, the algorithms for genome sequencing technology were originally made arose from microarray technology in the 1990s and 2000s, which arose from a lot of algorithms to quantify astronomy star difference. Because what happens is we do this bridge amplification process, and we, for every attached piece of DNA to this glass plate, we have a different fluorescent binder that chemically binds to nucleotides, and then for each vertical location, we take a picture of it and look at the fluorescent color. In this example, it's really clear that red can be G, yellow can be T, etc. And then we look at a sequence of these pictures, and for each pixel in the picture, assemble that into a string. So we can look at tens of millions to hundreds of millions of sequences massively multiplexed at the same time. And so the diagram doesn't show this well, but you can see that in these pieces of DNA, there's actually a adapter sequence of DNA that binds to the glass plate on either side of the read, so that both ends bind to the glass plate, and we read the DNA up until it, if we think about the biophysics, it's bending at the top, and it can't be read anymore with the imaging platform. So we actually have 2 strands of DNA with an insert gap between them. So we can think of this as a contiguous substring with a insert gap in the middle that's defined by these biophysical processes. So this is called paired end sequences. And then once we have these sequences, we often have 150 to 250 base pairs on each end of the paired end read. We can then go align that to a known reference genome, or assemble our own reference genome. So in our next class, we're going to cover the algorithmic basics for genome assembly. And Oxford Nanopore's technology, which I talked a little bit about last class, has this reader protein and a motor protein that's unzipping the DNA, pushing one strand through. Then we're putting an electric charge on this pore and reading the output with a microvoltmeter. And then we can either use a RNN or a hidden Markov model to take that squiggle and convert that into a string. So that's the sort of first practical application of deep learning in biology that we'll talk about. If you recall last class, we've gone and sequenced your DNA to see if you have a genetic disorder or risk for different things. We'll talk about polygenic risk scores in just a moment. We talked about how we take the string data and use last class and how we use concepts that Euler invented with Eulerian walks and cycles to view our strings as a graph, and we make a walk to assemble the shortest common superstring. I've changed our lecture plan slightly. Last class I mentioned that we would cover the shortest common superstring on LeetCode. We'll actually cover that in our next lecture. Are there any questions? If anyone has any questions, please feel free to raise your hand and I'll cover them as we're going, since this is a small class this semester. So unfortunately, things are more complex than that system I just described. Besides having different genes that can control things, we can have variants in genes that cause different alleles to arise in a population. Mass media would call a variant a mutation, and that these can arise through errors in copying genetic material, radiation, etc. This is caused by altered versions of proteins. It causes altered versions of proteins. So when we have an AT here, which changes to a GC, the protein that DNA is transcribed to mRNA, which is then translated by ribosomes. The secondary structure of the protein, how it folds, changes based on the different sequence, which can cause diseases or different functionality. A really interesting part of studying genomics and genetics today is how we think about so there's approximately 20,000 human genes, and genes are on average a few hundred to a few thousand base pairs in length of nucleotides. A base pair in computer science terminology would just be a character. We can think of this, a major area of research is studying genomes at a population scale. So let's say that we're doing a genome-wide association, abbreviated as GWAS, to look at height in populations. So we look at short people in a population, average people in a population, and tall people in a population. And so we follow all the ethical consent data to get their sequencing data. And so you may have noticed that a few weeks ago, the genome startup 23andMe lost effectively all of their patients' sequencing data. I think 7.4 million individuals. Can anyone in the class think about why it would be terrifying to have your genome sequencing data stolen by a hacker? There's no wrong answer here. Hackers stole the password information to download potentially all of their database of 7.4 million people, and they don't know how many people's sequencing data was stolen. And so a reason this could be terrifying is I'll actually walk through it in this example a little bit before I answer that question. So once we have gathered millions of people's of data, there's lots of government projects. The NIH, NHGRI, all of us consortium is sequencing 1 million unique Americans. The UK Biobank, I think, has 500,000 individuals in the UK. We can look for single variants that are associated, we can do a single variant association test with a candidate gene, a candidate variant of a gene to see if it affects height in this polygenic non-Mendelian universe we're living in. And the third point here is that genetics can be really complex. We can have things in biology that we call epistatic effects, and that one gene or one variant may just be a proxy for another causal variant. So then what we do is we do an association test, and then we plot the negative log 10 p-values into this plot here. And so this plot here is called a Manhattan plot. And it's called a Manhattan plot because if you look at it, it looks like the Manhattan skyline. So you can see that we have different chromosomes numbered from 1 to 21. And each location in this plot represents the index of the string of the genome, where a mutation could arise. And then we set a significance cutoff. Mohammed, do you recall what the standard significance cutoff is? It's 10 to the negative 5 or 8, right? Yeah. Mohammed used to be the TA for the course. And I didn't remember that off the top of my head. So the p-value is really, really low. In most of science, we look for something like under 0.05 or 0.01. But we have to be really careful with false discovery rate and multiple hypothesis correction statistically here. What we have to, if you think of just plotting a normal distribution and you do 1 million tests and you're looking at the tail being significant, just by probability, you'll have random things that will appear significant that are not. So we have to do a stringent correction to make that p-value even lower to say that these SNPs, so a SNP is just a mutation in the genome, also called a single nucleotide polymorphism, that they're significant and associated with the trait. And then, have any of you guys gone in to pick up your prescriptions at CVS and seen the little 23andMe or Ancestry.com box that has the cheek swab for genome sequencing that you can buy? Have any of you done 23andMe or Ancestry.com? So Dr. Hasseltine has. So besides showing your genetic ancestry and potential illegitimate cousins that you didn't know about, and a lot of these companies are making what are called polygenic risk scores. And a major focus of my research group is combining polygenic risk scores with multimodal imaging like MRI and CT scans to think about how we can improve them. So after we do this significance test, and oftentimes we'll have 2 dotted lines, one for the raw 10 to the negative 8 p-value, and then an even more stringent, so this is negative log, so as we go up, the p-value cutoff is being more stringent. For our FTR corrected hypothesis. We'll take this list of significant SNPs, and then we'll make a really simple mathematical model that says, based on these SNPs that our genome-wide association test has found to be significant, what is your risk of breast cancer in the next 10 years? And there's a lot of debate about the ethics of approving this in terms of FDA regulations, but 23andMe will sell you this test. So the terrifying science fiction story in my mind is hackers just stole the genetic test that allows you to compute these polygenic risk scores for 7.4 million people. What prevents an insurance company from stealing that data and denying coverage to people whose genome suggests they have a higher risk for certain cancers in the next 10 to 20 years? So the ethics of genome privacy is really, really important. There's a lot of other important ethical considerations in polygenic risk scores also. So earlier this year, my group sequenced 18,000 American women with diverse genetic ancestry to look at risk of breast cancer, say in the next 10 years. And we partnered with the hospital at UCLA to do this, and we discovered that the score that you go to the pharmacy and buy your polygenic risk score test on has been trained on 250,000 women who lived in Scandinavia. So we discovered that these risk scores don't work if your ancestry is either African or Asian. But if admixture occurred in the previous 25 to 50 generations, the polygenic risk scores still work. So then, after we make these risk scores in these genome-wide analysis studies, we can move on to our final phase. We want to do fine mapping, functional validation work in model organisms, and meta-analyses between many studies. So this is just looking at mapping traits in the genome. We can also study the transcriptome. So if you recall, in an earlier slide, I mentioned how DNA is bunched into these chromatin structures. And what happens is DNA unwinds, and then we have these transcription factors and cis-regulatory elements in the genome that transcription factors bind to. This is sequences of genetic information in our substring of the genome that these transcription factors bind to. And this is a lot of molecular biology. This is a computer science course. This is just all of the background knowledge that you'll need to understand the context for the algorithms that we're working with. And so these transcription factors bind to these cis-regulatory elements and initiate transcription. And what they do is they unzip the DNA, read it, and copy one strand of DNA to mRNA, where we're swapping our thymine with uracil. And then once we have our mRNA, we can actually use the same Oxford Nanopore, Beijing Genomics Institute nanoballs, and Illumina massively multiplexed adapter ligation with fluorescence sequencing technologies on our transcriptome. So now we start to become really interested in using dynamic programming algorithms, which will be a major 2 to 3 week focus of the course. So this is just a preview of that. So we can have an individual genome say we go discover a new species of toad while we're doing a field expedition and we're a biologist. We can go de novo assemble that, or say that we're studying human genetic diseases and we've collected the transcriptome from some interesting cancer patients, we can go align that to a reference genome. So how do we quantify the amount of, and so this is interesting because in the central dogma of biology, it states that DNA always flows to mRNA, always flows to the proteome. If we have something that's messing up our chromosome or affecting expression, we can have a gene that's in our genome that's not expressed that we won't see the downstream physical manifestation of. So here we measure aligning the transcriptome to the genome in terms of RPKM. Here I've shown 3 sort of different genes. And so in a gene, we have exonic regions and intronic regions. An exonic region, an exon, is gene that codes a protein, and an intronic region is gene that doesn't fold a protein. And our intronic regions, after expression in the mRNA, are spliced out. So what our gene looks like in the proteome, in the genome, doesn't always correlate what we see in the proteome because of these splicing complexes. So you can see here we have 2 red blocks with this black line between them. That represents an intronic region here. And so we have our red gene, gene A, our gene B, and our gene C, which are different links. And you can see that we have a standard, one is 600 bases, one is 1100 bases, and one is 1400 bases. So you can see that we have those aligned to the genome. Does anyone want to take a gander from their algorithms class about what algorithm we use to associate and line up these 2 strings? So if you think back to we'll cover some leak code examples of this too. When we're comparing large groups of strings, it's not computationally efficient if we think in terms of big O notation to compare every index against every other index. So we actually use dynamic programming to split this down into small recursive problems using either the Needleman-Wunsch algorithm to align strings or the Smith-Waterman algorithm to align substrings. So we'll spend actually a lecture covering each of those, where I'll actually have you read the papers from the 1960s and 1970s that Needleman-Wunsch and Smith-Waterman wrote. And then we'll spend part of an additional 3rd lecture discussing how these algorithms are deployed massively in parallel. So if I went, and my lab doesn't have a wet lab, but let's just pretend hypothetically I'm a biologist. If I went and got a sample, sequenced it, and sent it to Dr. Hasseltine at the Genome Center, I might get 700 million reads back. And I would have to run these dynamic programming algorithms 700 million times to quantify abundance. So what RPKM is we're just saying reads per kilobase of exon per million reads mapped. So we're just coming up with a number that normalizes the gene length, the number of gene reads that align to it, so that we can have a value of gene expression. So we would often call this a RNA-seq experiment. So after we run our sequencing, do our alignment, and we're gonna cover how this algorithmically works in terms of these recursive dynamic programming algorithms on paper to derive the simple cases of the Needleman-Wunsch and Smith-Waterman algorithm. When we apply this at scale, there's a lot of things we're doing to index these searches, like seeding and using the Burous-Wheeler transform, which is going to be the 3rd lecture in this series. There's tools that you can run on the command line that can do these 700 million alignments against a string in just a few hours. And we might do some live coding demos of that too. So essentially, what RPKM does, and I won't go over, I've shown the formula here, and that we have 1 million reads, and then we have the number of mappable reads in each gene, and then the length of the feature. So we're essentially just normalized, and then I've gone through the calculations. So what this allows us to have is a sort of gene abundance matrix. So say we are sequencing 4 mice with cancer and 4 mice without cancer, we can go look at all 20,000 genes, have a normalized value that we can quantitatively compare for each gene, and then look at a statistical enrichment of the set of genes in the mice that have cancer versus don't. And so this is just sort of computational processing of the data we have. Finally, very few people are using RNA-seq as a primary method of discovery today. RNA-seq was invented in 2008, or became widely adopted in 2007 and 2008, and is still used. Most groups today are interested in doing single-cell RNA-seq, and that this is really popularized by a company called 10x Genomics that had a really brilliant bunch of biochemists who realized that we can have a whole platform to sequence individual cells, that we use DNA to tag itself, so we don't need to change the sequencers in a genome center we use to process this. So if we have a brain here, we could sort of view bulk RNA-seq as different Legos. And then single-cell RNA-seq is we can look at the different colors of Legos or the different cell types and quantify how many of those cell types there are. And then we can look at spatial transcriptomics, where we can look at doing genome sequencing on top of a pathology slide slice. This is really hot with like 10x Visium platform. And then there's also, my group does a lot of spatial proteomics work using the Codex platform from Akoya Biosciences. And then if we assemble all of the layers of the Lego, we get a representation of our actual tissue. So what happens in single-cell sequencing? We have our tissue, all of our cells, and so in RNA-seq, we'll just, like, if we're looking at tumors in mice, we'll just take out the tumor, dissociate it, throw it in a blender, and then just get a gene expression value across all cell types. What we do in single-cell RNA-seq is we actually, so we get this average gene expression across all cells, and we don't have a view into cell heterogeneity. So what single-cell analysis does is we actually put each cell in a tiny detergent bead using a microfluidic device, and then we give each cell a unique DNA barcode, and then we give each molecule a unique DNA barcode, so that we can actually then pull all of the reads together and then computationally untangle where they came from. And so each cell type here will have a distinct expression profile, and this is where we start to get into some fun deep learning stuff. Sorry, machine learning stuff, not deep learning yet. Can anybody, have any of you guys taken linear algebra? Can any of you explain from a stats class how principal component analysis works? So we can look at breaking down data into principal components that contribute to the most and second most amount of variance to a data set. There's some nice analogies to decompositions like SVD and linear algebra. And what we can do is we have 20,000 dimensions of features, because each dot in this plot is a cell. And we're characterizing that by we want to show 20,000 features in 2 dimensions in a plot. So this is just a dimensionality reduction, unsupervised machine learning approach. And so if we can do this with PCA, but it looks awful. So we use algorithms like t-SNE, which we'll actually derive later in the class, and UMAP, which are like PCA, but we take away some of the linearity constraints so that we have nice grouping of cells. So the logic here is once we've taken away the linearity constraints in our decompositions or PCA, that a cell in the plot is more similar to a cell that it's closer to, but distance is meaningless. So an inch in the plot over here could be 3 inches in the plot over here. So we have this relative relation between embedding the cells, but the nonlinearity makes distance and distance metrics in this embedded space difficult to interpret. So why is single cell sequencing useful? Would anyone like to take a guess? So if we're looking at a disease, say cancer, there's a lot of different cell types all mixed together in tissue. We can have immune cells, we can have healthy normal cells, we can have tumor cells, and it's really useful to distinguish them. So an application where my research group has used single cell RNA-seq is we looked at, this is actually a bad illustration. We looked at melanoma in the skin, not lung cancer, and we did a needle biopsy of both blood and tumor. And then we used a technology called flow cytometry, where we essentially shine a laser at cells and shoot them through a tube to sort them out. So we only captured immune cells. And then we made this UMAP nonlinear unsupervised learning dimensionality embedding. We'll talk more about t-SNE and manifold projection in a future lecture, where we then made these plots where from tumor and mouse blood, we showed that each dot is an immune cell, and we could group and label different types of immune cells. Then what we were able to do is deep learning is all the hype these days. 90% of what my group does is deep learning, vision transformer models, large language models. There's oftentimes cases where you don't have enough data, where doing a more simple supervised learning approach is more appropriate. So here we just did L2 normalized logistic regression, and we're able to predict cells in blood, whether or not whether that immune cell lineage had previously been in tumor or not. Then we went to UCSF and validated this with 2 oncologists, Adil Daoud and Michael Rosenblum, and discovered that this is actually useful in the clinic. And we use this as a liquid biopsy for treatment prognosis about what immunotherapy you should get for certain types of melanoma. So oftentimes we can take a complex measurement from genomics, transcriptomics, single-cell genomics, single-cell transcriptomics, and do interesting algorithmic things with it post hoc. So next we can talk about proteomics. So unlike genomics or transcriptomics, we can't directly sequence a proteome right now. So this is something I'm working on, but currently the state of the art, using some nanopore technologies, some groups at the Riken Institute in Japan can distinguish one amino acid in a protein, but not the full sequence. So we can use what's called protein peptide mass spec, where we take a tissue, grind it up, get the proteins, and then I guess blast them with a really powerful laser, and then look at the spectra from that to quantify what is in a sample at a submolecule level. And so this is not accurate for sequencing. We can use it for some validation experiments and presence or absence of proteins. Where proteomics has really taken off in the past couple of years is work from Gary Nolan's lab at Stanford, where he invented the Codex platform, which he now calls the phenocycler fusion platform, where we take a pathology tissue slide and then can measure up to 100 proteins with fluorescent expression on the surface. So this is some work that I did in my postdoc where I made, I looked at some lung adenocarcinoma patients where we looked at immune cell infiltration into their tumors. And I made this color-coded scheme based on the protein markers. We could say red means the immune cell is infiltrating the tumor. This is a hot tumor. And so a new class of cancer drugs called immunotherapy, where we can actually activate the body's own immune system against itself, could be really useful here. We also have ones where some immune cells enter the tumor, and then some samples that we call a cold, where the immune system can't enter the tumor. So these patients have poor prognosis. We also can think about genome editing. This is a really hot topic, in that we can do a lot of screens and use technologies like CRISPR that use guide RNAs to potentially modify germline traits that cause disease. And I believe the first CRISPR therapeutic for certain genetic heart conditions was approved by Verve Therapeutics earlier this year, and the dose is around, the drug costs $70,000 per dose. So that's sort of a crash course in sequencing technologies, molecular biology, and genetics. So now I'm going to talk about how we're going to wrangle with this data in the course. And so I think Mohammed actually wrote this. We're not going to grade this exercise, but you should do it to fulfill your understanding of the material. So we talked about this process of, we're going to call this supplementary exercise. I'm going to post this like a homework. You just need to do it on your own time, but it won't be graded. So we talked about this process of how we can do a GWAS analysis to look for genes that are causally related to some disease, and then make a computational prediction algorithm using machine learning to make a polygenic risk score where we predict risk of disease. And so in this, to do a real GWAS study takes a few hundred gigabytes of RAM at least for a small case. So we've made a simple one that will run on a Google Colab notebook or your laptop. So let's imagine a fake scenario. You are working as a population geneticist for the government of a large country, trying to understand associations between a complex genetic trait, a phenotype, and genetic variants in a sequencing study conducted on hundreds of volunteer participants. In this study, there are 50 patients in the case cohort and 100 people in the control cohort. For these participants, 1,000 particular SNPs, this is way fewer than in the real world, SNP 1 through SNP 1,000, are measured and reported. In a real-world study, the number of SNPs tested can be several million. We call these SNPs either C alleles or T alleles. You are required to conclude whether there is significant evidence whether any of the C allele SNPs contribute to a person's risk of developing the complex trait. We were really mean in previous semesters in that we coded up a random number generator that made the distribution of SNPs uniquely tied to your UTA ID. And then we had a grading script that printed out the GWAS plot that your UTA ID generated, and to grade them, we made sure that they all matched. Since we're not grading this anymore, you just need to do this for your own enrichment. So the problem is you are working as a population geneticist for the government and trying to understand associations between a complex trait and the genetic variants in this study. So there's 50 people with the disease, 100 people without, and we're looking at 1,000 genetic variants. So this is a typo. This should be supplementary exercise 1. We'll give you a Python 3 script. So this is the beginning of our coding bootcamp. As a basic requisite, you need to be able to run Python 3 code on your laptop, and it's probably helpful if you can install RStudio and make that run too. This is like something that I would spend 10 minutes in a previous semester doing, but if you can't install RStudio, you can just go ask GPT-4, how do I install RStudio, and it will give you a nice step-by-step answer. If you're having trouble with installing any of the environments you need, I am available by office hours. So we're going to actually give you a script that will generate a unique code. We're not grading this, but this way you can, if you have friends in the course, you can try this with different seeds to make sure that you're getting the same result. And the first thing you want to do is look at how the artificially generated data set for our GWAS project is structured. So you'll get like, I think it's not an Excel file. I think we make this a CSV, comma separated value, or a TSV, tab separated value file, where we start at index 0 because we're computer scientists. So we index at 0. Give me 1 sec here. And then we have, in our case and control for that mutation, how many people of the 50 people who have the disease, how many people have that version of the mutation versus the other version of the mutation. So we've simplified the math here because population genetics and thinking about when we have a mutation, it's not just like you get a mutation and then everyone in the population has it. This is a lot of stochastic random walks that we model with partial differential equations to look at a mutation going to fixation or loss in the population. So we can look at our case here and say that 10 people have it and 40 people don't. So 10/50ths of the population have fixation, have this mutation that's trending to fixation through many generations of reproduction. So you'll run this script, it will generate this, and then the next test is you'll need to create odds ratio for the allele and then do a Fisher exact test. What is an odds ratio? The odds ratio is defined as the ratio of the odds of event A in the presence of B and the odds of A in the absence of B. The odds of an outcome are the ratio of the probability that the outcome occurs to the probability that the outcome does not occur. So we can say odds is P divided by 1 minus P. And so an odds ratio quantifies the strength of the association between the two events. So let's give an example here. Suppose there are two alleles, T and C, just like in this supplementary exercise. The number of individuals in the case group having allele T is represented by A, and the number of individuals in the control group having allele T is represented by B. Similarly, the number of individuals in the case group having allele C is represented by X, and the number of individuals in the control group having allele C is represented by Y. So we've just told you where all of those values are, and then we make a contingency table. And then So in this case, the odds ratio for allele T is A given B, meaning A to B, divided by X to Y, which in mathematical notation is A divided by B divided by X divided by Y. And so we calculate that for every SNP. In the real world, this is millions or hundreds of millions. And so the next test is Fisher's exact test. So Fisher's exact test is a statistical significance test used in the analysis of contingency tables. Depending on the type of test performed, left tail, right tail, or twin tail, it checks if there's a significant evidence that the odds ratio is bigger than, smaller than, or different from 1. You should download the SciPy library and use their built-in function, scipy.stats.fisherexact, to find out the p-values associated with each SNP. For further information regarding this, please refer to the documentation. So we also need to think about type 1 and type 2 error. Type 1 error, a false positive, is if this guy goes to the doctor and the doctor says, "You're pregnant." Type 2 error, a false negative, is if this very pregnant woman goes to the doctor and the doctor says, "You're not pregnant." We need to think about different ways to correct for type 1 and type 2 error in our data set, summed across all of these tests we're doing. This slide normally gets a lot of laughs too. A Bonferroni correction reduces type 1 but increases type 2 error because it's conservative. So taking this concept of type 1 versus type 2 error into our homework, we want to think about correcting p-values. So assuming each association between a SNP, supplemental exercise, since we're adding the journal club and not grading all of the homeworks. So assuming each association between a SNP and the phenotype is an independent hypothesis, we want our effective p-value to be 5 by 10 to the negative 8. So we ask you what the Bonferroni corrected p-value is. And so we've simplified this a little bit. So if we're doing this for a real population study, we'll have these indices for each chromosome. We've just given you 1,000 SNPs, so we're not indexing chromosomes. This is sort of like doing a test on a GWAS analysis on one microchromosome that we start at index 0 to 1,000, and then the values of the SNPs are determined by this random number generator based on your UTAID. And then you can look at setting your p-values for ones that fit the initial cutoff. And then we do our Bonferroni FDR multiple hypothesis correction, since this is negative log 10, to make a second line. And then the ones above it, after we construct our odds ratios from the sequencing results, run our Fisher's exact test, do our Bonferroni correction to sum all these p-values. So you can see that there's quite a bit of scripting you'll have to do to complete this assignment. This is why this is the programming boot camp this year. This is, I'd say, a pretty average amount of scripting you'll have to do for an assignment. You'll have to install the SciPy library, use file.io to parse the CSV file the random number generator makes, go through each SNP, calculate the value, use a plotting library like Matplotlib and Seaborn or ggplot and calplot. I don't care what plotting library you use. Most of the assignments are designed to be in Python 3. Matplotlib and Seaborn is a pretty good, we'll do some examples of plotting libraries, is a pretty good option there. You're also free to take the data from Python and do this in ggplot2 and its associated libraries if that's something you're interested in. And then we have the SNPs, or the mutations that are associated with disease in the population that we can then go and make a polygenic risk score that 23andMe uses. So this is a simplified example of actually implementing from scratch the algorithms that population geneticists use to study genetic traits in a population. And so that's the first supplemental exercise. I'll give around 15 minutes in future lectures to go over results with anyone who codes this up on their own time if they want to, and TAs will also be available for that. So in a real world GWAS, millions of SNPs are included as opposed to the 1,000 in this assignment. And that so you can see a diagram here of SNPs in different chromosomes proven to have significance for different traits taken from the GWAS catalog. And we often use this GWAS catalog result to look for and aggregate data sets to study new polygenic risk scores. And so these SNPs are located at different loci on the chromosome. And so for the Manhattan plot, in this last plot, I've shown, and these coding exercises, this coding exercise is designed to take roughly 1 to 4 hours to complete. So going through that is an excellent interactive way to study for questions that will be on the midterm. If you do these interactive coding assignments, you're learning the material that we're going to ask you about on the midterm if you choose not to do the extra credit project assignment. So in this plot, I've shown a real GWAS analysis versus our mock one that we've computationally scaled down so that it can run on any laptop computer and doesn't have constraints. So I'm going to assign that this week. I'm also going to, I'm actually going to introduce I'm actually going to delay the introduction of our next supplementary assignment. So just doing these as we go, we're only meeting once a week. If you give yourself 1 day a week to cover the journal club stuff and do a little bit of code for either the supplementary exercise or homeworks, you'll be in a great spot to get a good grade in the course. We're going to do in next lecture, we're going to cover genome assembly algorithms, and we're going to give you another fun supplementary exercise. So after graduation, we're going to pretend you've joined the CDC rapid response team, and that we're going to actually give you some genomes to assemble that are Yersinia pestis, the bacteria that causes the Black Death. And there's a patient that has it, and your boss wants a plot showing the distribution of gene lengths in the genome, but unfortunately the genome is not assembled. So I'm actually going to take a quick moment to introduce our journal club paper for next class. So when we're reading academic journals, I'll post a link to this. For peer-reviewed journals, if you're not at a university, these cost money to access. So if you're on UTA Wi-Fi or the UTA VPN, all of the links we send for journals should be accessible by the UTA library. So I'm on UTA Wi-Fi right now, so if I go click on this PDF, it will download the paper that normally costs money, and it'll put this watermark on that says I downloaded it through UTA. So this paper has been cited 5,000 or 6,000 times. Give me 1 second. I'm just going to close the door really quick. So this paper has been this paper has been cited 5,000 or 6,000 times. And in the next class, we're going to go over implementations of de Bruijn graphs. There'll likely be some conceptual questions about de Bruijn graphs on the midterm if you choose not to do the extra credit. But so a de Bruijn graph is an efficient representation of the shortest common superstring problem. So next class we'll actually move into our LeetCode modules, and I'll do some real-time solving of LeetCode strategies for the shortest common superstring problem. I believe that's a LeetCode hard. I'll talk about the design patterns for all the master students in the class that are studying for FANG interviews. And then we'll talk about the how a normal de Bruijn graph is, and then I'm going to assign you guys this short paper to read. I believe it's a applications note in Oxford University Press Bioinformatics, and it's only, I think, 3 pages. So I'll send you this paper. You'll read it. We'll discuss it. That will be your attendance grade, the discussion of it next class. And then you're actually in supplementary exercise number 2 going to run this algorithm on your laptop to assemble this Black Death Yersinia pestis genome. Okay, moving. Are there any questions? So I'm actually going to push this to next class. So we just talked about genome sequencing. So when we're doing genome sequencing, in a bacterial genome, like this Yersinia pestis, the bubonic plague genome that we're going to be assembling next class, a bacterial genome is a fully circularized contiguous piece of DNA. So we can say that our ideal is to get this fully circularized double helix, and that bacteria can have really tough cell walls, as well as some plants. So we often need to use an enzyme or chemical to break down the bacteria and use nasty chemicals like chloroform to extract the DNA before we sequence it. There's a pretty standardized set of, when we're doing genome assembly in practice. So today I'm just talking about this is in the context of our introduction to programming file I/O. I'm going to spend less time on this than in past semesters. When you move to being a professional researcher or work in a company, ChatGPT has really, and GitHub Copilot, have really changed the game. My best PhD students have become 10 times more productive since ChatGPT came out, because previously, if you were working on a project on computer vision and then you switched to working on a sequencing-based project, you'd have to go to Stack Overflow, read all the documents, be like, how do I read in a FASTQ file? What's a FASTQ file? Go to Stack Overflow, Google it, and figure it out. Now, simple file I/O and data formatting based things can be fully automated with large language models. And when you're coding for the class, we're not going to ask you on any of the rated test silly questions like code file I/O, because in the real world you're going to be using GPT-4 to do that. So you should be doing that in your coding for the class also. But just to go over this really quickly, we normally have a metadata string, which is so we see that we have in a text file for each read groups of 4 strings and 4 lines, and that these repeat themselves. So the first one is a sequence identifier with information about the sequencing run, and the exact contents of this line vary based on what genome center you use. Number 2 is the actual sequence. Number 3 is a separator that's just an empty plus line there for historical reasons. And then we have these encoded Phred scores, which are the confidence inside the genome sequencer that the Illumina machine had on its output. And so we talked about how on the chemistry for the plate for Illumina, we have adapter sequences on both ends. So we have this inner distance that we can't sequence. So we end up having an adapter, the first half of the read, this gap, and then the second half of the read, and then the adapter. So when we're sequencing, we're actually sort of treating this as two separate reads from different ends of this, and then not bridging it over fully. Okay, let us, for the second assignment that we'll go over next week, I've made a list of some of the tools we want to talk about. So this slide is one of my favorite slides. I give this to all new PhD students in my lab. I call this a computer scientist view of stats. If we're not doing something advanced like Bayesian-inspired deep learning questions, most of stats can be broken down into a pretty simple, if you're writing a paper towards a biological or human health audience, this is sort of a little cheat sheet. And on the top axis, it shows us, is your data categorical, ordinal, or numerical? And do we want to do a parametric or non-parametric test? I'll go over how to put that in a second. And then we have different columns. Or do we want to measure data? Do we want to describe population data? Do we want to compare one sample to known values, et cetera, et cetera? And then in each cell, it gives you the name of the test to use. I'm going to post all of the slides onto Canvas for each lecture. So if you're stressing about taking photos of this, you'll have the full resolution copy also. It gives you a little diagram of what the plot should look like in your paper or assignment. So this is an incredibly useful resource. We can talk about parametric versus non-parametric test. Non-parametric test can be used on ordinal and nominal scale data, though also on interval and ratio scale data. Can be used on small samples. Most importantly, can be used on data that are not normally distributed, can be used where the samples are not selected randomly, and always have less power than the equivalent parametric test. We use parametric tests mainly on interval and ratio scale data. They tend to need larger samples. It tends to need to, data should fit a particular distribution. We can do a transformation to make it fit to a distribution as a pre-processing step. Samples should be drawn randomly from the population, and it's always more powerful than the non-parametric equivalent. So we can think about distributions and skewness also. So if we're looking at any type of biological data, we can have a symmetrical distribution where the mean equals the median equals the mode. But then we can have distributions that have tails too, where we can have most of the data fitting in a distribution, and then we can have a long tail of the distribution where data is in the extreme. And we can oftentimes mathematically reason about this as actually adding two distributions together to get a skewed distribution. An important concept is the t-test. We're not going to go over much of this. Have any of you guys had a college stats course? Okay, so a one-sample t-test, is there a difference between a group of people and a population? An unpaired samples t-test, is there a difference between two groups? And then a paired samples t-test, is there a difference in a group between 2 points in time? And my cheat sheet should go over where to use these. Yes, it does, with the unpaired and paired t-test. And then we can also use a z-test when the population is large or when population variance is known. So last semester, when we actually assigned the GWAS homework that you guys are doing, a lot of people struggled with how to implement in their code a Bonferroni corrected p-value. And so to move the line of which SNPs are significant up. So to get the Bonferroni corrected p-value, you just take the original p-value and divide that by the number of tests performed. And as we talked about, Bonferroni reduces type 1 error, but increases type 2. And then these were so just one last stats point. We can think of a p-value as the probability of obtaining test results as extreme as the observed results given the null hypothesis. So being extreme can sometimes mean to have test statistics being more than a value, a right-tail test, or sometimes being less than a value, a left-tail test. Other kinds of extremes also exist. The null hypothesis is the hypothesis encapsulating the idea that there is no guarantee to repeat the observed result, a statement of no difference. If the p-value is small enough, we can confidently reject the null hypothesis and conclude that there is statistical significance associated with our extreme observation. In GWAS, we usually use the chi-squared test for finding the p-value, and we talked about how to implement the p-value. Implement the test using the SciPy libraries earlier in the slides. So a lot of the course is tailored for the master's students and undergrads and computer scientists who are thinking about FANG interviews. So we're going to cover a lot of functional programming paradigms in Python that will make you more efficient in your leak code practice and tech interviews. Python is nice because if we think about the evolution of programming languages in the past 20 years, oftentimes we're becoming more and more abstracted away from the machine. And that a low level, we can have assembly language, which is super low level, or we can have C and C++, where you have to manage memory at a low level. At the other end of the extreme, we have a lot of functional programming languages, like Scheme, Haskell, etc. And then we have very functional, like object functional languages like Scala that can compile to Java bytecode, but are still functional. Functional languages were very, very popular in theoretical computer science. In some cases, like Haskell, for example, has some nuanced challenges in that there is no concept of a variable that can be modified, and that every variable has to be a argument to a recursive function that is called over and over, hence the name a functional programming language. But math and logic is really, really clear in functional programming languages. It's really easy to move from a proof to a code and say this line of the code corresponds to that line of the proof. So around 10 years ago, many of the proprietary trading firms on Wall Street, like Jane Street, Millennium, and hedge funds, switched to coding most of their trading strategies in functional programming languages because the languages could be evaluated by a trader who is not the coder. And likewise, a lot of these clear understanding that we can gain from insights into functional programming are useful for coding efficient research code and can cut down the number of lines we use when solving a FANG tech interview, for example. So a theme of the course is going to be to introduce some of these programming paradigms, both in our bioinformatics assignments and in our prep for industry. So the first one is a list comprehension, which is a structure inspired by Haskell that is implemented in Python 3, where we say in green, do this, x plus 1, for this collection in this situation. And then if we put that in brackets, this will generate the list that creates. So here are some examples. We can say, let me one sec here. That just messed up. Give me one second again. So here I've opened just the Python REPL next to our assignment. So let's say our first list that we define here, can everyone see that all right, is L equals I for I in range 10. And so then if we look at what L is, it's every, Python is 0 indexed. Every number between 0 and 9. So we can also add an if condition. We can say L equals I for I in range 10. Our first one, if I modulo 2 equals 0. Can anyone tell me what this line of code will change our output? Can anyone recall what the modulo operator does? So the modulo operator checks divisibility. So in our if condition here, if whatever number for the data that we're doing something on is evenly divisible by 2, modulo 2 equals 0, we return it. So we take the elements in our first list and return it in our second list. So how these constructs of list comprehension came around is how languages are evaluated. So functional languages have lazy evaluation. So this means if I was going and coding something in Haskell, I could go and define a list of 1 to infinity, and it's only when I'm evaluating a value or using a value in that list for a downstream task that the value will be calculated. So I can go, say, make a list of 1 to infinity, and Haskell will define that. It has to be in a recursive function since it's a functional programming language. And so some of these evaluation strategies, the syntax for them became really popular and led to use of things like list comprehension. So we can also do if else. So we can say I for I if I modulo 2 equals 0, else I times I for I in range 10. Does anyone want to take a crack at what this line of code is going to do? So what's changing between our first example and our second example? We've just added this else I times I line there from the second one. So all that's going to change, I messed up my syntax. That should have been an equal, as we're not doing a variable assignment there. So all that's changed is it creates this list, and then if the value in the range we're looking at is not divisible by 2, it goes to the second case condition, where it's the value times itself. So 0 times 0 is 0, 1 times 1 is 1, 2 is divisible by 2, 3 is not divisible by 2, so we return the else condition, 3 times 3, so on and so on. We can also do a nested comprehension. You can do ij for I in range 10, for j in range 10. And so you can see what this list generates, and I didn't assign that, so I just put that out, is all possible values of I and j in those two subsequent ranges. And so where this is really useful is if you need to populate data for a coding interview question during your technical interviews. This is about as efficient in terms of number of characters you can type, and it makes your code easier to debug quickly, as you don't have to go through many for loops. We can also do a dictionary comprehension, so I can say, can anyone share what data structure a Python dictionary is based on? So a Python dictionary, we can define an empty dictionary. A Python dictionary is Python's super efficient implementation of a hash map, which is just key value pairs. So I can say, in my dictionary, the value of Jacob is my age. And then I can go look at my dictionary, and it's a key value pair. And that we can efficiently go and retrieve those super quickly in sublinear time because of the data structure of the hash map. And so this is really useful for associating things like patient data. And I can use a list comprehension to actually construct a dictionary. So I say for I in the range of 0 to 9, make the key have a value of the number times itself. So 0 is 0, 1 is 1, 2 is 4, 3 is 9, etc. So list comprehensions are incredibly useful. So that structure we just covered is a dictionary in Python. Dictionaries are unordered collections of unique values stored in key value pairs. Dictionaries, lists, and sets are sort of the workhorse data structures in Python that we'll use for a lot of assignments. So we can see that D of A is, we use these curly brackets and then colons to separate groups of key value pairs. So key A is 10, comma, key B is 20, and we can retrieve these. So it's important to note that dictionaries are unordered. The items in them are stored without any index value. They're unique. Keys in dictionaries are unique. I can't put a second key of A in. If I did, the background behavior in Python would just replace the value, and they're mutable. Unlike a functional programming language, we can add, modify, and remove key values after creation. And so We are all familiar, so I'm going to now talk a little bit about the string matching problem. So we are all familiar with the general idea of string matching. We use string matching in such applications as internet searches, word finds in text editors, and even in DNA pattern matching. So we assume that text is an array T of size 1 to n of length n, and that the pattern is an array P of length 1 to m of length m, where m is less than n. We also, that's not a bit shift, that's a typo. We also assume that the elements of P and T are characters of the finite alphabet sigma. For example, sigma equals, our alphabet is A and B. We want to find P equals AAB, and T equals this long string. So, for example, we are given a long string, A, B, A, B, 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 A, etc., and the pattern A, B, A. In a brute force method, we compare T and P at each letter. And so the idea of string matching is that we want to find all occurrences of the pattern P in a given text T. And we could use the brute force method for string methods, but this is a terrible time complexity. So in some of our LEAP code in the next class, I won't actually cover this today. We're going to talk about string matching, how the Robin-Karp algorithm works, and then move on to dynamic programming approaches for string matching. And so to end today's lecture will be slightly shorter than previous lectures since we are starting our journal club next week and our coding. So this is actually a bit longer than normal lecture. How class is going to be structured is we're going to start with our journal club, then do our lecture and coding. And so there's no live coding today. We'll start that in journal club next week. But I wanted to give you some historical context about sequencing. Sanger sequencing is a approach that we can start with a DNA sequence and essentially copy it with these special nucleotides that have fluorescent colors, and then measure this for one sequence at a time. And this is still used for some validation experiments, but it doesn't scale to the massively multiplex 700 million sequences we can sequence at a time with Illumina. I believe Sanger won a Nobel for this. So with Sanger's method, we extract and purify genomic DNA, we fragment it, we make a clone library, we sequence those clones, align sequences, and close the gap. The cost per megabase, or 1,000 nucleotides, is thousands of dollars, and it takes time. But this method was used to sequence the first genomes. So the first viral genome was completed in 1977. The first organism was completed in 1995. The first eukaryote was completed in 1996. Yeast, that Nature paper was done by my undergrad advisor when she worked at the Venture Institute. And then the first multicellular organism was done from 1998 to 2002. And then we finished the first plant in 2000. The Human Genome Project completed in 2007. They published competing Nature and Science papers the same week. There was a public and private version of this. Craig Venture's company, Celera Genomics, used Sanger sequencing, and it cost $70 million, which competed with the NIH's competing project. Using Roche 454 sequencing, about a year later, James Watson's genome cost $2 million to sequence. This slide is actually outdated. When I made this slide last year, it cost $1,000 to sequence the genome. Using the latest Illumina PacBio platform, the amortized cost is down to $200,000 $200 for a 30x coverage genome. So we've gone from sequencing single genes to complete genomes, single transcripts to whole transcriptomes, single organisms to complex metagenomic pools, and model organisms to anything. So that concludes lecture for today. If there's any questions, I'm happy to chat after class, and we will fix the Canvas link promptly after class today. Fantastic. So we're all done for today. I will send it to you. That's what I just mentioned.
All right, we've gotten a little done. I've contacted some people that I'm trying to find out if we know anybody at that company.
Oh.
My brother Bill doesn't, but he's in the wrong generation, so I've emailed somebody else.
Okay. I'm really grateful for your...
Well, I don't know if we'll get anything done, but I do know someone at the Mass General, and I do know my brother would know somebody at the Jimmy Fund. You're already starting the process here, so, I mean, we're doing more than one prong here.
Yeah, I think it's been a... We can walk and
Sure, I
talk.
have, unfortunately, I put my car in the Netterman.